In this video, I'm going to give a general overview of viral replication cycles, and then we're going to discuss more in depth the two different bacteriophage replication cycles. So in general, because viruses are not considered to be alive, we want to avoid saying life cycle. So instead, I'm going to be using the phrase replication cycles, because viruses, once they get into a host and within the correct cell, they are able to replicate and make more viruses. And so this process is going to be referred to as the replication cycle. Now in this class, we're going to look at both bacteriophage, which only infect bacteria, and animal viruses. And it turns out there's a lot of similarities actually between the two replication cycles, but there are also some key differences. So this video is going to focus more on bacteriophage replication cycles, of which there are two. And then in the next video, we'll focus on animal virus replication cycles. So let's go ahead and get started. So there are two different replication cycles that a bacteriophage might use. Now, it's not that they just get to decide which one they want, okay? Some viruses can only do the lytic cycle, and some bacteriophage do the lysogenic cycle. So with the lytic cycle, notice the lytic sounds like lysis, which means to basically blow up. And so during the lytic cycle, the bacteriophage is going to infect a bacteria, take over the cell, make lots of new bacteriophage, and then get out, okay? So it's very efficient. Infect, replicate, exit. And it is going to kill the bacteria in the process because it's going to lyse its way out, or it's going to cause the cell to undergo lysis. So let's take a look. So the first step is the bacteriophage is going to attach to the host. Remember, we talked about host range already. So the bacteriophage will attach to receptors on the host cell. So here's the bacteriophage. Notice its genetic material is in blue. Here's the bacterial cell, and its genetic material is in pink. So once the bacteriophage attaches, the next step is called penetration, because here, the bacteriophage is going to inject its viral DNA into the host, okay, much like a syringe. Now, what the next picture doesn't show you is sometimes this leads to the host DNA getting chopped up into little bits because the phage doesn't want to compete with host processes, right? It wants to take over. And so as long as the phage DNA is the only DNA, then it can take over, no competition. So once the phage DNA is inside, that's when it starts to take over. It's going to use DNA polymerase. It's going to use RNA polymerase, the ribosomes, all the things. Okay, it's going to take over the host, and it's going to make new viral DNA and new capsid proteins. And because this is a phage, it's also going to make more sheaths and tail fibers. So that's what you see here, okay? So we call this step biosynthesis because the cell is making all the things the phage needs in order to get out and infect new hosts. Now, y'all, please know, like with phage, that's another word for virus, but we use phage when we're talking about bacteriophage, the viruses that infect bacteria. Okay, but notice in this picture that right now, the phage DNA is not inside the capsids where it belongs, okay? So that's actually the next step, which is called maturation. And this is where the new intact phage kind of spontaneously assemble. All the things come together to have an intact virus particle. And so these are now ready to get out and go infect new bacterial hosts. So the last step is lysis. So notice they're kind of like punching their way out of these host cells. And that's the host cell is essentially being blown a bit, okay? So make sure you know the name of each of these steps and the gist of what is happening. Now, before we move on to the lysogenic cycle, I want to stop for a minute and remind you of a process called transduction. It was a type of, or it is a type of horizontal gene transfer. And remember, horizontal gene transfer is how a bacteria might acquire bacterial DNA from a neighboring bacteria. And with transduction, that was when a virus is involved, okay? And so remember I mentioned here at this second step, sometimes when the bacteriophage injects its DNA, it causes the host DNA to get chopped into little bits. And that's important because those little bits of DNA are still scattered within this cell. So as the lytic cycle continues and more of the capsid and the sheath and the tail fibers are all made, and they might be made individually, okay, just for simplicity, they kind of showed it all connected together in this picture, but they can all be kind of separate. And so what happens is when maturation occurs and all the parts spontaneously assemble and come together, sometimes chopped bits of host DNA, so the pink DNA, will accidentally get packaged. So then when that bacteriophage that's carrying bacterial DNA gets out, it's going to go and infect another bacteria. But instead of injecting viral DNA and causing this whole cycle to repeat, it'll inject bacterial DNA, which does not harm the bacteria. Okay, so this is where transduction comes into play. When it goes through maturation, sometimes it accidentally picks up bacterial DNA instead of viral DNA, which it then will inject into a new host. All right, so notice this bacteriophage, when it's going through this cycle, is very efficient. It attaches, it makes new virus particles, and it gets out, okay? This next bacteriophage replication cycle is called the lysogenic cycle. And this one, it has those same steps, but there's kind of a middle step, okay? And I call it the integrate and chill step. So let's take a look. So the first step is always going to be attachment and penetration. So notice the bacteriophage has attached to a bacterial cell. It is injecting the phage DNA into the bacterial cell. Now, rather than chopping up this DNA to bits, instead, it is going to let its DNA integrate into the hosts, okay? So now it's just one big old piece of DNA. The bacteria doesn't know what has happened. It can't detect that. And so this bacteria is just going to keep copying its DNA and dividing like it normally would. And this, so see here is where this bacterial cell has undergone binary fission. So it eventually copied its DNA and stretched and split into two. So notice each of these cells now has viral DNA. And this process is going to continue going on for a long time, okay? So I only show it happening once, but these two cells will then divide, and now we'll have 4 cells that have viral DNA. They'll divide, and we'll have 8 cells with viral DNA, okay? So this is the integrate and chill phase. Now, eventually, these bacterial cells are going to find themselves stressed out, right? Life always gets stressful. And when that happens, the viral DNA does not want to be part of a stressed out host, so it gets out. The viral DNA is removed from the host, and then it just finishes that lytic cycle. So notice it's going through biosynthesis. It's making more viral DNA. It's making more viral capsid sheaths and tail fibers. They're going to spontaneously assemble during maturation. So now we have intact virus particles that are ready to get out. They do. They leave by lysis. So notice, we still have attachment and penetration. We still have biosynthesis, maturation, and lysis. Okay? There's just an extra step with the lysogenic cycle. And think about why this is. How is this advantageous to the bacteriophage? Well, do we agree? I'm going to go back one slide. Let me get through all these animations first. Do we agree that here, one phage infected one bacteria, and at the end of the day, it released from one single bacteria? Well, with this method, now each of these cells has viral DNA. So by the time these cells get stressed out, there might be hundreds or thousands of bacteria that all have, unknowingly have, this viral DNA. So when the whole population gets stressed out, suddenly we have thousands of bacteria that have virus particles being made within them. And then around the same time, all these thousands of bacteria are going to die, and they're going to release that many more virus particles into the environment. Okay, so this is just like a sneaky way of essentially infecting lots of other bacteria just by integrating into their DNA and chilling for a while. Okay. All right, y'all, that's the end of this video. Remember, both the lytic and lysogenic cycle are just for bacteriophage, those viruses that infect bacteria. In the next video, we're going to focus on how animal viruses replicate. All right, as always, let me know if you have any questions.